Let's start with question 1. Part A, Part E. Here we have a pyramid of energy, and we need to identify where organisms in trophic level 1 get their energy from. Remember, trophic level 1 always contains the producers. These are plants and other photosynthetic organisms. They get their energy directly from the sun through the process of photosynthesis. The sun is the primary source of energy for almost all ecosystems. And producers capture this energy and convert it into chemical energy that flows through the food chain. So your answer should be the sun through photosynthesis. Now Part A, Part 2, asks us to calculate the percentage of energy transferred between trophic levels 2 and 3. Looking at the numbers, trophic level 2 has 1,500 kilojoules per square meter per year, and trophic level 3 has 120 kilojoules per square meter per year. To find the percentage transfer, we divide the energy at level 3 by the energy at level 2, then multiply by 100. So 120 divided by 1,500 equals 0.08 times 100 gives us 8%. The question asks for one significant figure, and 8 is already one significant figure. So the answer is simply 8%. Always remember to show you're working in calculations like this. Part A, Part 3 asks us to state 2 reasons why energy decreases between trophic levels 1 and 4. This is a very common question in ecology. Energy decreases because organisms use energy for their own life processes. First, energy is lost as heat through respiration. Whenever cells respire, heat is released. Second, energy is used in metabolic processes like growth and repair. Other valid reasons include energy lost in movement and muscle contraction. Not all parts of an organism are eaten by the next trophic level. Energy is lost in feces through egestion, and not every organism in a trophic level gets eaten. You only need to state any 2 of these. Part A, Part 4 asks us to describe the advantages of using a pyramid of energy compared to a pyramid of biomass. A pyramid of energy is actually more accurate for several important reasons. First, it gives a more accurate representation of energy transfer and loss through the ecosystem. Second, it accounts for different rates of reproduction among organisms. Third, it accounts for different lifespans. Fourth, it accounts for different sizes and body compositions of organisms. Fifth, it accounts for time and seasonal variations. And finally, it allows for better comparisons between different ecosystems. You only need to describe any 2 of these advantages for full marks. Remember, biomass pyramids can be misleading because they don't show the rate of energy flow. Moving to Part B, Part I. We need to complete a sentence defining the term population. This is a basic definition you must know. A population is a group of organisms of one species living in the same area at the same time. The 2 key words here are area and time. So the completed sentence reads, a population is a group of organisms of one species living in the same area at the same time. Make sure you remember these key terms for your exam. Now Part B, Part 2 asks us to describe and explain the shape of the graph at point X. This graph shows what happens when a new species is introduced to an ecosystem. At point X, we see the classic S-curve or sigmoid growth pattern. Let me explain what's happening. First, the population increases rapidly because birth rate exceeds mortality rate. There are plenty of resources available, and there's no competition or predation initially. Then, the rate of population growth starts to slow down. Why? Because resources become limited. As the population grows, competition increases. Predators may arrive, and diseases can spread. Eventually, birth rate and mortality rate become more equal, causing the population to plateau. This plateau represents the carrying capacity of the environment. So in summary, rapid growth due to abundant resources, then slowing growth due to limiting factors, finally stabilization when birth and death rates equalize. Now we move to question 2, Part A, about the distribution of rods and cones in the human retina. Looking at the distribution pattern, we can identify several key differences. First, overall there are more rods than cones in the retina. Second, cones reach their maximum density at the fovea, which is at 0 arbitrary units on the graph. Third, rods are most numerous on either side of the fovea, not at the center. Fourth, there are actually no rods present at the fovea itself. This is why we see better in bright light at the center of our vision. Fifth, outside the fovea, the number of cones remains relatively constant. And sixth, there are more rods on the left-hand side of the fovea compared to the right. For your answer, you need to describe any 4 of these differences clearly. Part B asks about nocturnal animals compared to animals active during the day. Nocturnal animals are adapted for night vision. They have more rod cells or a greater proportion of rods in their retina. Why? Because rod cells are much more sensitive to light and work well in low light conditions. They help animals see in darkness. An alternative way to answer is to say that nocturnal animals have fewer cones. Why? Because color vision, which cones provide, is less important when it's dark. Both explanations are correct as long as you link the structural difference to the functional advantage for night vision. Part C has 3 short parts about the iris response to bright light. Part I asks for the name of this response. It's called the pupil reflex. When light is bright, the pupil constricts or gets smaller. Part 2 asks which effector contracts during this response. The effector that contracts is the circular muscle of the iris. When this muscle contracts, it makes the pupil smaller. Part 3 asks for the type of action shown by the paired effectors. The answer is antagonistic. This means the circular muscle and radial muscle work in opposition. One contracts while the other relaxes. This is the same principle we see in other muscle pairs in the body. Part D shows a diagram of a synapse between 2 neurons. Part I asks us to label A, B, and C. Looking at the diagram, A points to the small spheres containing chemicals. These are vesicles. B points to the receiving area on the second neuron. This is the receptor protein. C points to the chemical messengers being released. These are neurotransmitters. For Part 2, we need to explain how the neurotransmitters, which is Part C, move across the synaptic gap. They move by diffusion. This means they move randomly from an area of higher concentration to an area of lower concentration. So after being released from the vesicles, they naturally spread across the gap to reach the receptors on the next neuron. This is a passive process that doesn't require energy. Question 3. Part A shows a stage in plant reproduction after pollination. Part 1 asks for the name of structure X. Looking at the diagram, X is the pollen tube. This tube grows from the pollen grain down through the style toward the ovule. Part 2 asks us to describe the events after this stage that lead to fertilization. First, the pollen tube continues to grow and eventually enters the ovule through a small opening. Second, the male nucleus travels down this pollen tube. Third, once the pollen tube reaches the female gamete, the male nucleus fuses with the female nucleus. This fusion is fertilization itself. Make sure to mention that this happens within the ovule. After fertilization, the ovule develops into a seed. Part B, Part I asks us to suggest reasons why a plant species might use self-pollination instead of cross-pollination. There are several valid reasons. First, there might be a lack of pollinators, no insects, birds, or wind to carry pollen between plants. Second, the plant might be isolated with no other plants of the same species nearby. Third, self-pollination uses less pollen and less energy because the plant doesn't need to produce showy flowers or nectar. Fourth, it increases the chance of successful fertilization and offspring production. Fifth, If the plant is already well adapted to its environment, maintaining the same genes through self-pollination can be beneficial. And finally, it guarantees reproduction, even when conditions for cross-pollination are poor. Any three of these reasons would earn full marks. Part B, Part 2, asks us to discuss the potential effects if a plant population only uses self-pollination. This is about the consequences of no genetic exchange. First and most importantly, there will be less or no genetic variation in the population. All offspring will be genetically identical or very similar to the parent. Second, this means the population is less likely to survive or adapt to environmental changes. If conditions change, all individuals will be affected similarly. Third, there's less resilience against disease. If one plant gets sick, all can easily get sick, and diseases spread quickly through genetically identical populations. Fourth, this increases the risk of extinction. Fifth, harmful recessive genetic diseases are more likely to appear and spread. Overall, the population's long-term survival is compromised without genetic variation. Part C reminds us that meiosis produces gametes and is one source of genetic variation. But the question asks us to state two other sources of genetic variation in populations. The first is mutation. These are random changes in DNA sequence that create new genetic variants. The second is random mating or random pollination. When individuals pair up unpredictably, it creates new combinations of genes. Another valid answer is random fertilization, the chance combination of which sperm fertilizes which egg during reproduction. Any two of these are acceptable. Remember, genetic variation is essential for evolution and adaptation. Part D asks us to describe mitosis and its roles in organisms. Let me explain mitosis clearly. Mitosis is a type of nuclear division. During mitosis, a cell divides to produce two genetically identical daughter cells. The chromosomes are first copied, then they separate equally into the two new cells. The chromosome number in each daughter cell is maintained. So if the parent cell had 46 chromosomes, each daughter cell also has 46 chromosomes. Now, what are the roles of mitosis in organisms? First, growth. Mitosis allows organisms to increase in size by adding more cells. Second, repair. Mitosis replaces damaged tissues like healing a cut. Third, replacement. Mitosis replaces old or dead cells throughout your life. Fourth, asexual reproduction. Some organisms reproduce entirely by mitosis. And fifth, production of organs and tissues during development. So mitosis is essential for growth, repair, and maintenance of multicellular organisms. Part E asks a straightforward question. Name the unspecialized cells that divide by mitosis. The answer is stem cells. Stem cells are unique because they haven't yet differentiated into specialized cell types. They can divide many times by mitosis through a process called self-renewal. And they have the potential to develop into various specialized cells like muscle cells, nerve cells, or blood cells. This is why stem cells are so important in medicine and development. Question 4, Part A asks us to describe what is meant by the term excretion. Excretion is often confused with digestion, so be careful. Excretion is the removal of waste products of metabolism from the body. Metabolism refers to all the chemical reactions happening inside cells. These reactions produce waste that must be removed. Excretion also includes the removal of substances that are in excess of the body's requirements. For example, the lungs excrete carbon dioxide, which is a metabolic waste. The kidneys excrete urea and excess water and ions. The skin excretes water and salts and sweat. So remember, excretion is about removing metabolic wastes and excess substances. Part B asks us to state the name of the substance excreted by the lungs. The main answer is carbon dioxide, written as CO2. Carbon dioxide is produced during cellular respiration and must be removed from the body. The lungs also excrete water vapor, so this is also accepted as a correct answer. So either carbon dioxide or water vapor is fine. Part C shows a simplified diagram of a kidney cross-section, and we need to label P, Q, and R. Looking at the diagram, P points to the outer region. This is the cortex. The cortex is where filtration begins. Q points to the tube leading out of the kidney. This is the ureter. The ureter carries urine from the kidney to the bladder. R points to the inner region. This is the medulla. The medulla contains the collecting ducts and loops of Henle. So remember, P is cortex, Q is ureter, R is medulla. Part D asks us to describe and explain the difference in composition of fluids in structures X, Y, and Z. Let's go through each one carefully. Structure X contains blood. Filtration occurs between X and Y, where small molecules are filtered out. Proteins and blood cells are too large to be filtered, so they remain in the blood. Structure Y contains glomerular filtrate. This fluid contains glucose, water, urea, and ions. These are the small molecules that pass through the filter. Structure Z contains urine. Notice that Z has no glucose. Why? Because all the glucose has been reabsorbed back into the blood. The body doesn't want to lose glucose. Z has a higher concentration of urea because water has been reabsorbed. Some water and ions have also been reabsorbed back into the blood. So Z is more concentrated and contains urine. This whole process shows how the kidney filters blood, reabsorbs useful substances, and produces concentrated urine for excretion. Part E asks us to describe two ways amino acids are processed in the liver. The liver plays a crucial role in amino acid metabolism. Process 1 is deamination. This involves removing the nitrogen-containing part from amino acids. The nitrogen is converted into urea, which is then excreted by the kidneys. Deamination allows the remaining carbon skeleton to be used for energy. Process 2 is assimilation. This means the liver converts amino acids into proteins that the body needs. These proteins include plasma proteins, clotting factors, and many others. So in summary, the liver either breaks down excess amino acids through deamination or uses them to build new proteins through assimilation. Question 5. Part A, Part 1, shows an evolutionary tree diagram. We need to suggest visible features that developed at points A and B. At point A, which is on the branch leading to mammals, the features that developed include hair and external ears. Hair is a defining characteristic of mammals, and external ears or pinna are also mammalian features. At point B, which is on the branch leading to birds, the features that developed include wings and feathers. These are adaptations for flight and are unique to birds. So for A, answer hair or external ears. For B, answer wings or feathers. Part A, Part 2 asks us to identify the two groups that share the most recent common ancestor in the diagram. Looking at the evolutionary tree, we need to find where two branches split most recently. According to the diagram, apes and rodents share the most recent common ancestor. This means they diverged from each other more recently than from any other group shown. So the answer is apes and rodents. Part A, Part 3 asks us to suggest which groups have the most similar and least similar DNA base sequences compared to crocodiles. Looking at the evolutionary tree, birds are closest to crocodiles on the tree. They share a more recent common ancestor. Therefore, birds have the most similar DNA sequences to crocodiles. Sharks are farthest away on the tree. They diverged much earlier. Therefore, sharks have the least similar DNA sequences to crocodiles. So the answer is most similar to birds, least similar to sharks. This shows how DNA similarity reflects evolutionary relationships. Part B asks us to describe the structure of a DNA molecule. This is a fundamental concept in biology. DNA has two strands or chains wound together in a double helix shape. Imagine a twisted ladder. The DNA molecule contains four types of nitrogenous bases: adenine, thymine, cytosine, and guanine, often written as A, T, C, and G. These bases pair in a specific way. Adenine always pairs with thymine, and cytosine always pairs with guanine. This is called complementary base pairing. The base pairs are held together by hydrogen bonds. So in summary, double helix, two strands, four bases with AT and CG pairing, and hydrogen bonds between the bases. An annotated diagram would also be acceptable for full marks. Part C asks us to outline how the base sequences in DNA control cell function. This is about the central dogma of molecular biology. First, the base sequences in DNA determine the sequence of amino acids in proteins. This happens through transcription into mRNA and then translation. Second, amino acids join together to form proteins through protein synthesis. The order of amino acids determines what protein is made. Third, the sequence of amino acids determines the protein's three-dimensional shape, and the shape of a protein determines its function. For example, enzymes have specific shapes that allow them to catalyze reactions. Neurotransmitter receptors have shapes that allow them to receive signals. Hormones have shapes that allow them to trigger responses. So ultimately, DNA base sequences control cell function by determining which proteins are made and what those proteins can do. Question 6, Part A, Part 1, presents a clinostat experiment with maize seeds. We need to complete several sentences explaining the root growth. Let me read through the complete answers. First, auxin is produced in shoot and root tips. Auxin is a plant hormone produced at growing points. Second, the auxin travels down the root by the process of diffusion. It moves from cell to cell. Third, as the clinostat rotates, the effect of gravity on all sides of the root is equal. The clinostat rotates continuously, so no one side experiences more gravity than another. Fourth, this causes the distribution of auxin in the root to be equal. Because gravity affects all sides equally, auxin distributes evenly. Fifth, auxin stimulates cell elongation, causing the roots to grow horizontally. Since auxin is evenly distributed, all sides elongate equally, so the roots grow straight out sideways instead of bending downward. This experiment beautifully demonstrates how plants normally respond to gravity through uneven auxin distribution. Part A, Part 2 asks us to predict what happens when the rotation of the clinostat is stopped. Once rotation stops, gravity will no longer be equal on all sides. The lower side of the root will experience more gravity. This will cause auxin to accumulate on the lower side. Since auxin inhibits cell elongation in roots, remember it's different from shoots, the lower side will elongate less, and the upper side will elongate more. This causes the root to bend and grow downwards toward gravity. So the prediction is, roots grow downwards or towards gravity. Part A, Part 3 asks us to state the name of the tropic response to light. This is phototropism. Shoots grow towards light, which is positive phototropism, while roots usually grow away from light, so the answer is phototropism. Part B asks us to state and explain the expected appearance of cells from a plant that has wilted. When a plant wilts, it's because it has lost water. So let me describe what the cells look like. First, the cells are flaccid, meaning they're no longer turgid or firm. In severe cases, plasmolysis may occur. This is when the cell membrane pulls away from the cell wall. There's a lack of turgor pressure inside the cells. Turgor pressure is what normally keeps plant cells firm and supports the plant. The volume of the cytoplasm and vacuole decreases as water leaves. There's decreased or low water potential inside the cells. All of this happens because of a lack or loss of water from the cells. When you see a wilted plant, this is exactly what's happening at the cellular level. Thank you.